For any living organism to grow and divide, cell division is essential. This cell division results in the growth of the organism as well as maintaining a constancy of chromosome number in the daughter cells or in the next generation. During cell division, the major thing that happens is the division of the chromosome. All the body cells their functions are controlled by the chromosome. The chromosomes in eukaryotic cell are present inside the nucleus. Each chromosome contains one long DNA molecule which is wrapped around small proteins called histone proteins. These histone protein and the DNA altogether constitute chromosome. Initially in a cell, chromosome is present in the form of fiber called chromatin fiber. But during cell division, condensing and coiling of the chromatin fiber occurs, which results in formation of chromosome. This time the chromosome picks up stain. Chroma means color, zome means body. Hence they are called chromosome. Chroma means color, zome means body. The chromosome discovery credit goes to Walter Fleming, and in 1882 in salamander, an amphibian, he has discovered the presence of chromosome. He saw that the chromosome divides rearrange during cell division. The first cell division is mitosis. Mitosis means filament. Other cell division is meiosis. Each chromosome is formed after condensation of chromatin reticulum or chromatin fiber. Each chromatin material composed of nucleic acid, DNA, deoxyribonucleic acid and histone about 60%. Then the molecular structure of DNA. This was first studied by Rosalind Franklin in 1953 and later elaborated and properly discovered by Watson and Crick, for which they got the prestigious Nobel Prize in 1962. DNA are the macromolecules, means they are larger in size. They consist of many micromolecules, that is called nucleotide. So this is a nucleotide. Many such nucleotide constituted DNA. Nucleotide consists of pentose sugar, nitrogenous base and phosphate. So here is the double helical model of DNA. Here two nucleotide strands are wrapped around each other in a double helical pattern. Between that on the strand present the sugar and the nitrogenous base are posed into the gap. Two nitrogenous bases of each facing strand are joined with each other by a hydrogen bond. The nitrogenous base are adenine, guanine, thymine and cytosine, and adenine and thymine linked by double nitrogen bond, guanine and cytosine linked by triple nitrogen bond. They altogether form a ladder-like arrangement. Here it is presented, ladder-like arrangement, adenine, guanine, thymine, cytosine. Here is the hydrogen bond. Now move on to the structure of histone protein. Histones are basic proteins which helps in coiling and packaging of DNA into structural unit called nucleosome. A long stretch of DNA coiled around 8 histone octamer. For example, see 1, 2, 3, 4. At the back side imagine 1, 2, 3, 4. So 8 octamer around which a DNA is coiled 1.75 times to form one nucleosome. If we study the structure of chromosome, see this is a DNA. These are all histones. 8 number of histones. So the DNA now slightly coiled around the histone and gradually more coiling occur, and this results in the formation of nucleosome. Many such nucleosome coiled over each other, super coiled over each other, ultimately forming the chromatin fiber. The chromatin fiber further condense and coil to form a chromosome. It is just like a wool is creating a sweater to wear. So now the nucleosome, every human body, the 2 meter, each cell contains 2 meter long DNA, but the nuclear diameter is 6 micrometer. So proper packaging is quite essential for this 2 meter DNA to be present within 6 micrometer radius nucleus. The entire chromatin fiber is coiled and supercoiled and form the chromosome. Now basic points are, there are two types of nucleic acid, DNA and RNA. The component of chromatin, DNA and histone. The component of nucleotide, nitrogenous base 2 type, adenine, guanine, nitrogenous bases, pyrimidine, thymine, cytosine, uracil. Here say the nitrogenous bases are basically of 2 type, double ring purine, single ring pyrimidine. Purine example adenine and guanine, pyrimidine example thymine, cytosine, uracil. Here you notice one adenine always pairs with one purine always pairs with one pyrimidine, like adenine pairs with